How does a complex organism such as a human develop from a single cell, a fertilized egg, into the vast array of cell types such as nerve cells, intestinal cells, and bone cells that make up our bodies?
A stem cell is a non-specialized cell that can divide without limit as needed and can, under specific conditions, differentiate into specialized cells, which make up all the different cell types of our body. Stem cells are divided into several categories according to their potential to differentiate. The first embryonic cells that arise from the division of the zygote are the ultimate stem cells. These stem cells are described as totipotent because they have the potential to differentiate into any of the cells needed to enable an organism to grow and develop. The zygotic division produces blastomeres, which later make up the hollow sphere known as the blastocyst. Cells migrate within the blastocyst to locations that will later define the structure of the embryo and consequent organism. In this process called gastrulation, three germ layers arise: the endoderm, mesoderm, and ectoderm. These cells that develop from totipotent stem cells and are precursors to the fundamental tissue layers of the embryo are classified as pluripotent. Cells in these three layers will give rise to different parts of the organism. The endoderm eventually becomes the gut. The mesoderm develops into muscle, the skeletal system, some organs, and the connective tissue. The ectoderm differentiates into the nervous system and skin. These cells then become slightly more specialized and are referred to as multipotent cells. A multipotent stem cell has the potential to differentiate into different types of cells within a given cell lineage, or a small number of lineages, such as a red blood cell or white blood cell.
When a cell differentiates or becomes more specialized, it may undertake major changes in its size, shape, metabolic activity, and overall function. Because all cells in the body, beginning with a fertilized egg, contain the same DNA, how do the different cell types come to be so different? The answer is analogous to a movie script. The different actors in a movie all read from the same script. However, they are only reading their own part of the script. Similarly, all DNA contain the same full complement of DNA, but each type of cell only reads the portions of DNA that are relevant to its own function. In order for a cell to differentiate into its specialized form and function, it need only manipulate those genes and thus those proteins that will be expressed, and not those that will remain silent. The primary mechanism by which genes are turned on or off is through transcription factors. A transcription factor is one of a class of proteins that bind to specific genes on the DNA molecule and either promote or inhibit their transcription. Stem cell research aims to find ways to use stem cells to regenerate and repel cellular damage. Over time, most adult cells undergo the wear and tear of aging and lose their ability to divide and repair themselves. In a laboratory setting, it is possible to induce stem cells to differentiate into specialized cells by changing the physical and chemical conditions of growth. Researchers have recently developed induced pluripotent stem cells, or iPSCs, from mouse and human adult stem cells. These cells are genetically reprogrammed multipotent adult cells that function like embryonic pluripotent stem cells. They are capable of generating cells characteristic of all three germ layers. One of the most exciting possible uses of stem cells is in bioengineering new organs. For example, studies have been conducting using soluble factors in order to differentiate iPSCs into nephron cells. Using the NCBI gene database, we can find different genes, such as TGFA, which are responsible for these factors. Thus, scientists can determine the best possible microenvironment to make nephron cells and potentially a whole kidney.